Metallo crown ethers as potential drug delivery agents. As we were discussing, any drug which needs to be administered into the body is loaded upon the metallo crown ether compound and it easily enters the cell membrane, and this is how crown ethers work upon their drug delivery action. Moving on to the discussion of metallo enzymes. Metallo enzymes, as the word goes, are those enzymes which contain a metal ion. Many metallo enzymes are present in the body which have a variety of roles to play. Some examples include oxaloacetate decarboxylase, carboxypeptidase, carbonic anhydrase, and many others. So, we first discuss the mechanism of action of oxaloacetate decarboxylase. For that, we first need to understand what is this enzyme. This metallo enzyme is involved in the conversion of oxaloacetic acid to pyruvic acid, which is one of the steps in the Krebs cycle. Krebs cycle meaning the final process in carbohydrate metabolism. So, as we see, carbohydrate metabolism is a part of, if we consider it, then organic chemistry. So, all the branches are interlinked. The role of metal ion is present everywhere, including the biological system. As the reaction goes, which is mentioned over here, the removal of carbon dioxide is the decarboxylation step in the above conversion, which is catalyzed by oxaloacetate decarboxylase. This is the reaction in which there is removal of carbon dioxide from oxaloacetic acid. The reactant is oxaloacetic acid, which is producing pyruvic acid, and carbon dioxide is being released in the system, and this is releasing a lot of energy since it is the final process in carbohydrate metabolism. And as we all know, carbohydrate metabolism produces a lot of energy in the system, and this reaction is catalyzed by oxaloacetate decarboxylase. This enzyme, oxaloacetate decarboxylase, contains zinc ion, which is present in the biological system. So, as we can see, again the role of metal ion coming in the enzyme. Then there is another enzyme which is carboxypeptidase, which catalyzes the hydrolysis of polypeptides. This is a polypeptide chain, as we can see on the screen, and hydrolysis of the polypeptide chain, which results in two products being formed. Carboxypeptidase is a zinc containing enzyme and is characterized as bovine carboxypeptidase A, which is isolated and crystallized and studied by X-ray analysis. It has been found to contain 307 peptide units and one zinc ion which is present per molecule. The zinc ion which is present in a pocket in the folded chain of the enzyme. The enzyme is a macromolecular species in which zinc ion is one of the enzymes, just like it was present in oxaloacetate decarboxylase. Zinc enzyme is also an important constituent of carboxypeptidase enzyme, and carboxypeptidase enzyme contains 307 peptide units, and in those 307 peptide units, which are folded in the form of as we can say an apple, which has pocket above, and in that pocket the residence of zinc, and that place is called the active site of the enzyme. So, we can imagine carboxypeptidase to be in the form of an apple and the whole is occupied by zinc ion. The enzyme is very specific in its action. That is, it cleaves the peptide bond at the carboxylate terminal end only. It cannot cleave the peptide bond from any other side. Secondly, it hydrolyzes only those substrates where the R group is either an aromatic or a branched chain aliphatic group. When we see the structure, in the peptide, the R should be aromatic group or a branch chain aliphatic group, and the cleavage, as we can see, occurs from the C terminal or carboxylate terminal end. This is the structure, as we can see, of the enzyme carboxypeptidase, which is also called as bovine carboxypeptidase A. Carboxypeptidase is a macromolecule which exists in a folded form in the biological system. Zinc ion resides in the depression on the surface of the molecule, as I gave the example of an apple, and you can see the zinc ion which is residing in the depression, and the place where the zinc ion resides is called the active site of the enzyme. That is, this is the place where the substrate, that is the peptide, gets attached, which needs to be cleaved or which needs to be hydrolyzed. A pocket develops below the active site to accommodate the peptide. The peptide, that is the substrate which has to be hydrolyzed. Since the role of the enzyme is to hydrolyze the carboxylate terminal end of the peptide, the enzyme can self-cleave its own carboxylate terminal end. To prevent this, the enzyme keeps its carboxylate end tucked away from the active site. So, the active site now has only the peptide carboxylate terminal end which needs to be cleaved. The enzyme itself has number of carboxylate terminal ends, because ultimately the enzyme is also a protein. So, it also contains a number of carboxylate terminal ends, but its own carboxylate terminal ends are tucked away or kept away from the active site, so that it cannot cleave its own carboxylate terminal end. When model studies of the enzyme have been carried out, that means outside the biological system, the enzyme has been studied. The bovine carboxypeptidase enzyme has been studied, and the enzyme activity has been studied using cobalt 3 plus as the cation instead of zinc 2 plus. It has been found that the enzyme has retained its activity once this happens. Cobalt 2 plus has not been used in model studies. If we are assuming that there is zinc 2 plus which is present in the enzyme, we can take cobalt 2 plus instead of zinc 2 plus, because both of them are transition elements, both of them have almost similar properties. But instead of cobalt 2 plus, cobalt 3 plus is chosen because of the absence of protective protein layer around it. Cobalt 2 plus can easily get oxidized to cobalt 3 plus. So, outside the system there is no protective protein layer which can oxidize CO2 plus to CO3 plus. Now, whenever the carboxypeptidase A enzyme acts, that is, it cleaves the peptide units, a lot of energy inside the body, then there are certain conformational changes which occur in this enzyme. Now, BCA, as it's called, or bovine carboxypeptidase A, is a pancreatic enzyme. It exists or is produced in the pancreas. When the system gets positioned on the enzyme for hydrolysis, or when the substrate is positioned, the following interactions take place. That is, when the peptide which needs to be hydrolyzed is positioned on the enzyme, there are following changes, following interactions in the enzyme which take place. That is, number one, the carbonyl group of the substrate coordinates to zinc ion, replacing the water molecule. That is, the substrate, which is the peptide unit, contains carbonyl. It replaces the water molecule of the enzyme and gets attached to zinc. The side chain of the amino acid to be cleaved is enveloped by a hydrophobic pocket. Next, the arginine residue 145. Arginine residue 145 means that the residue or the amino acid residue at the 145th position moves through 200 picometers to come within the hydrogen bonding distance of carboxylate group of the substrate. Another change which happens is the tyrosine 248, that is the tyrosine amino acid in the 248 position, moves through 1200 picometers to hydrogen bond to the NH of the amide group through its phenolic hydrogen. These are the conformational changes which happen in the enzyme while it is hydrolyzing the peptide. A glutamate group also interacts with the substrate through a water molecule. The water molecule becomes polarized and it attacks the carbonyl of the amide, resulting in the cleavage of the peptide linkage. So these all interactions takes place, all changes takes place within the carboxypeptidase A and the peptide unit and between them also. Now, since we have discussed now the chemistry of two enzymes, Metallo enzymes, now we come to the chemistry of biochemistry of very important ion, which is essential for our living system, is the biochemistry of iron. Iron, we all know, is an essential constituent of hemoglobin. And how does it work? That is what generally we don't have a full understanding, and which is explained in the biochemistry of iron or the bio-inorganic chemistry of iron. And how does it work? Iron is a metal ion having a very important role in the biological system. It is contained in hemoglobin, which is the oxygen carrier molecule in higher animals. By the meaning higher animals, I say that it includes animals as well as human beings. Lower animals contain different type of iron carrier proteins. Heme is termed as a chromoprotein. Chromoprotein means a protein which is attached to a prosthetic pigment group. Pigment means colored. Heme consists of a porphyrin ring which is formed by 4 pyrrole nuclei joined by methine bridges. This is the structure of heme. As we can see, iron is in the center, and iron which is in the center is in the form of iron 2. There are 4 pyrrole rings. Pyrrole means 5-membered nitrogen containing rings, as you can see on the 4 corners of iron. Iron is 4 coordinatively associated with 4 nitrogens of the 4 pyrrole rings. These pyrrole rings are joined together by methine bridges, and the pyrrole rings contain associated groups like CH3, CH2, COOH and CH double bond CH2. This is the overall structure of heme. Without the globin part, globin means the protein which is surrounding the heme. So as we can see, this is a 4 coordinate structure. So in the description of the structure, we say that it is a planar 4 coordinate structure. All the 4 pyrrole rings and Fe2 plus are in the same plane in oxyhemoglobin. The 4 pyrrole rings and Fe2 plus. The 4 pyrrole rings, as you can say, and the Fe, they are in the same plane as in case of oxyhemoglobin. Now, what do we understand by oxyhemoglobin? In the structure of heme, there is, as we can see over here, it is a 4 coordinated structure. When we consider the structure of hemoglobin, where the globin part or the protein part surrounds this heme, there is a 5th coordinate position, that is, iron has a maximum coordination number of 6. So a 5th coordination position is occupied by one of the amino acids from the globin part which is surrounding this whole structure, and the 6th position is occupied by oxygen in the case of oxyhemoglobin. So, as we can say, oxyhemoglobin, when the fifth coordination position is occupied by nitrogen atom from histidine of the protein surrounding the heme or the globin part, and the sixth coordination position is occupied by the oxygen molecule, and this is the state in which hemoglobin carries oxygen. Then we have deoxyhemoglobin, when the sixth position, coordination position, is occupied by weakly bonded water molecule and not carbon dioxide, as a misconception. We all know the color of heme, and this color of heme, it arises out of a charge transfer transition to low lying pi and pi star orbitals of the conjugated porphyrin ring from the metal ion. Whenever electrons are transferred from one orbital to another, that is from a low lying low energy orbital to a higher energy orbital, and when it there is a momentarily charged transfer, it produces an intense color in the protein molecule, and which is blood red, as we all know, the color of heme. And this kind of a charge transfer is a momentary transfer. Now, the role of the globin part, that is the protein part which is surrounding the heme unit, is that the iron 2 which is present in heme has been found to be susceptible to oxidation, and it forms iron 3 easily, because iron 2, as we know, the configuration is D6, iron valence shell configuration is S2D6, so iron 2 will be 3D6 and iron 3 will obviously be 3D5. So, from iron 2 to iron 3, the change is very easy, because from D6 to D5, which is a half filled state, the change becomes very easy as the molecule of iron tends to become more stable. This can happen by the oxidation by oxygen in the presence of water. That is, the presence of water is absolutely necessary for the oxidation of iron 2 to iron 3, and iron 3 is of no use in carrying oxygen, since iron 3 is a D5 system, and a D5 means a stable system, and it can undergo no reaction, it cannot carry oxygen. Hence, the role of the globin part is to provide hydrophobic protection to the heme molecule and prevent the oxidation of iron 2 to iron 3. So, globin part prevents water molecule to come near the heme part and prevents the oxidation, because of course oxygen has to combine, then only heme can carry oxygen. When we discuss the structure of heme exists as a tetramer. There are 4 heme groups which are attached to 4 globin molecules. That is, heme does not occur as a singular structure, instead it exists as a tetramer. 4 heme groups, 4 globin molecules in tandem, in association with each other. There are 2 alpha chains containing 141 amino acids and 2 beta chains having 146 amino acids in heme. When hemoglobin, the prior role, the majority role of hemoglobin is to carry oxygen, and there is a certain mechanism which hemoglobin follows in carrying oxygen. This mechanism was given by Peretz, and this is explained on the basis of structural changes which occur in the hemoglobin molecule. In deoxyhemoglobin, that is when in the 6th coordination position water molecule is attached, iron 2 is not in the same plane as the 4 pyrrole nitrogens. When we saw the structure of hemoglobin, as we can see over here, we had said that iron 2 is in the same plane as the 4 pyrrole nitrogens in case of oxyhemoglobin, But in deoxyhemoglobin, when the 6th position is occupied by water and not by oxygen, that means when hemoglobin is not carrying oxygen or when it has released oxygen in the tissues, at that state iron 2 and the 4-pyrrole nitrogens, they are not in the same plane. It is no longer a planar structure, and as a result of this, there is a configuration which is T2G4Eg2. It has unpaired electrons. How this configuration occurs? Since water molecule is attached in the 6th position to iron 2, iron 2 means D6 configuration, and D6 means with water as a weak molecule, the electrons tend to remain unpaired and they obtain a T2G4Eg2 configuration. And as a result of this, since the electrons are unpaired, the size of iron 2 is very big and it cannot fit into the slot of the 4-pyrrole nitrogens. And as a result of this, the tips, that is the nitrogen atoms, are pulled upwards towards iron 2 because of coordination, and this results in a structure termed as the tense structure of deoxyhemoglobin. As a result of the above coordination, a distorted pyramidal geometry exists, and this structure is thus labeled as a tense structure because it's a pulled up structure. It is not a relaxed structure. So this is all for today. Thank you so much.